Hello, everyone, and welcome to today's webinar, FAIRR 101, Computational Tools for FAIRR Analytics. Emily is a part of the customer success team aiding in advanced customer technical use cases. She recently defended her master's degree in computational biology at the University of Toronto, specializing in machine learning for single-cell genomics and protein-protein interactions. Emily, thank you so much for joining us, and I'll pass it over to you.
I want to begin by first highlighting components of life science workflows that are often seen and some pain points that might accompany those components. First, I want to begin with a user poll to kind of gauge the experiences of our audience today. Which of these have you experienced during a computational project? A project works on one machine but not on another, getting results that make you suspect you are missing part of the code for an analysis, or spending a large amount of time trying to find the correct data. Feel free to cast in your vote if you haven't done so already. So the results that I'm seeing on my end are that a large number of the audience has voted for the first option, that a project will often work on one machine but not on another. And I also see a lot of audience members have the experience of having some difficulties finding the correct data. And I definitely agree with the audience. I think those are pain points that I myself have experienced. So let's go over a quick visual. If we can end the poll, we can go over a quick visual of what that exactly would look like. So in a life science workflow, the start to finish, the overall high level would be going from data to discovery. You have some data that you would like to analyze. Maybe you got it off of Geo, Zenodo, or it was some acquired data, and you want to extract some information to make a nice plot, like you can see on the discovery end. But of course, specifically in computational analytical life science workflows, that's not usually a direct path that you can do. There's usually some intermediaries in the middle from data to discovery. So for example, to actually understand the information to extract from the data in a computational life science workflow, you often need some code. This might be some R code, some Python code, some MATLAB code that you use to process the data in order to get to the discovery. But usually code cannot be run out of the box. You often also need a configuration or an environment or a compute resource to actually run this code to process the data to get to the discovery. And the visualization of this process as a linked path is also really important, because when you actually lose one link in the path, you will not get to the discovery, which we can go over right now. If, let's say, configuration is grayed out, you don't know what dependencies you need to begin a project, or more importantly, you're inheriting a project from a past team member and you don't know the specific versions you need, well, of course, you can't use the data and the code, even if you have those available, and you can't get to the discovery. If the link were to break that code, maybe you're missing some utility functions, you're missing files altogether, it doesn't even matter if you have the data already. You will not have the full configuration to run the code and the data to get to the discovery. And like many of the audience members highlighted that they experienced, if you can't find the data, even though it's the beginning, the linked path is still holding intact. You can still have the code and configuration all set up. But if you don't know the data type you need, you can't get access to the data, or you don't know the correct version of the data to use. Maybe it's something like DetMap. You don't know exactly the version to use. The faultiness in the workflow will trickle down, and you will not be able to get to discovery. So now that we've kind of highlighted the components of the life science workflow that are really important, with respect to FAIRR and the FAIRR principles, how can we exactly highlight the advantages that need to be present in each component? So, highlighting by FAIRR, code has to be accessible. This might result in answering the questions: where is the code? What is the repo name? Who do I ask for access? What does the code do, and how do I run it? The full code base has to be accessible in order to actually run the analysis properly end to end. Data in a life science workflow has to be findable, and you need to be able to answer the questions: where is the data? Who owns the data? How do I get access? Has it been processed? And if there are multiple copies, what version do I use? An environment or configuration has to be interoperable. What packages and specific versions are needed? How do I install them? What kind of GPU or how much memory? And what operating system do I need to use? Can this only run on Windows? Can this only run on Linux? Can this only run on Mac OS? And abiding by FAIR principles, this is kind of an outline of the typical tenets of each component that we would like to see in order for the full life science workflow to be present and to be able to make that progress from data to discovery. And so there are some tools existing to kind of combat this reproducibility crisis that we see in life science workflows and improve the abidance by FAIR, which is findable, accessible, interoperable, reproducible, and reusable in the life science workflows. So the first one that we can highlight is a tool like Docker for the environment. Docker has a lot of benefits when it comes to being able to make your workflow fair. It allows you to containerize your entire application or your entire workflow as an executable or as an interactive virtual environment. You can enter a built Docker container that will contain all the necessary configurations to run a designated application. When building a Docker container to run as an interactive virtual environment or as an executable, the goal of Docker is to allow for pre-configured dependencies, which can avoid for any end user to experience clashes or unsolvable errors when it comes to package installation. And lastly, Docker is customizable. The ability to create layers within a Docker container are made available via package managers, and it allows you to add all the necessary packages within your Docker container for full operation. That way, Docker containers can be run out of the box, once again, designed to run out of the box for a specific designated application, which really allows you to set up your application or your life science workflow for full runnability. When it comes to providing code and abiding by FAIR principles for code, we can think of a tool like GitHub. You can really use any Git provider, but we would like to highlight GitHub as it's one of the more recognized Git providers in the life science or computational space. When it comes to making your code available on GitHub, there are many benefits. It allows you via committing and pushing to provide traceable progress of your project from start to finish, and now allow you to provide a roadmap of the progress that was made in a project and the changes that were made in files along the way. GitHub has collaborative configurations for different versions of your code base via branch, allowing you to create different versions of the code base within the same repository or within the same central hub that allow you to avoid merge conflicts or allow you to avoid differing versions of the code base. And lastly, GitHub is a very portable format for sharing your code. You may have seen GitHub links linked very frequently in high-impact journal publications, and it allows your code to be available open source for full release and re-implementation capabilities. And lastly, when it comes to data, there are several well-known life science or computational science repositories available. Ones that come to mind may be the Geo repository, Zenodo, and potentially any acquired data that you may have within your organization. When it comes to data repositories, the benefits include a direct download of numerous choices of life science source data. Geo and Zenodo, they have APIs, they have FTPs. There are many different ways that you can download a data set from Geo or Zenodo. When it comes to repositories like Geo, there's formatting and standardized formatting ready for analysis. We see examples of FASTQ files in Geo or maybe some other gene count expression matrices. They're formatted in a familiar way that allows you to readily use them for analysis. And lastly, specifically in a repository like Geo, there's often important metadata within a given data set. This might include metadata about organisms or the extraction method for a given data type or time of extraction or maybe biological context. There's metadata that can be used in the experiment to provide biological context for the results. While these tools are available to combat the FAIR crisis or reproducibility crisis that we see in life science workflows, sometimes there are some common challenges that can arise in their use. So let's take Docker for an example. We highlighted the benefits of using Docker in your life science workflow, including containerization, pre-configured dependencies, and customizable layers and package managers. What are some practices that could lead to disadvantages with using Docker? Well, if an author publishes a Docker container, but there are some more complex packages that require extra steps beyond the container, installing those specific packages could be difficult or complex to understand their installation, and that could cause some hindrance in actually running the full Docker container. When it comes to using operating systems, sometimes installed packages will install further utility packages in the background depending on your operating system. And it can make some packages invalid or unfindable when the Docker container is transferred. For example, if a Docker container is built in Mac OS, it might install some additional packages that are only relevant to Mac OS and are unfindable on Linux. And lastly, while Docker is fully customizable and designed to allow you to avoid build clashes or unsolvable dependencies, if package versions are not saved, the build can easily go out of date and load newer versions of packages. Packages that come to mind for me are TensorFlow and PyTorch that have regular updates with functions being deprecated or being changed or being added. And so without saving specific package versions, just by the build going out of date, clashes that previously did not exist can suddenly appear. When it comes to providing code via GitHub, we have the advantages of traceable, collaborative, and portable. So what are some disadvantages that can arise? Even though GitHub provides you a way to trace your progress of your project from start to finish, sometimes GitHub is only used at the end as a means to provide the code base for publication, and it's used as a code dump, which could cause some issues. Even though GitHub has collaborative configurations, there might be still some instances of merge conflicts in the original version of the code base, which from the end user perspective could lead to some questioning of, well, what exactly is the correct version of the code base? Because there's been multiple collaborators on the same version. And while GitHub is a very portable format to allow you to provide your entire code base for release and reimplementation, sometimes the code can be present, but it's not exactly confirmed to be set up for optimal execution. And lastly, when we look at data through life science repositories. While it provides you a method for doing direct download of numerous choices of life science source data, repositories can be easily deleted, deprecated, or files are removed due to being out of date. Like I mentioned before, if you're trying to access a specific version of DepMap, if it's incredibly old, it may not be available on the original repository as it was the time the analysis was first done. While Geo, Zenodo, and other formats of data can be formatted in a standardized format ready for analysis, issues can arise if there's multiple versions of the same data with various levels of batch effect or biological noise, leading to once again the question of what is the correct version to use. And while the original data repositories may have conserved information built in to track the metadata of the contents, if that is not found outside of the repository, when it comes to reuse, it can be difficult to determine the type of data that is designed for that project, which I've also experienced myself. So that leads us to a question of, we have all these tools, we know these pain points, how are we going to use them? And that leads us to the question of, why would we build a tech stack, which is the topic of today's webinar? We begin with the base layer, with the topic of well-known pain points in life science workflows can cause a crisis from the path from data to discovery. It leads to an intermediary of existing tools provide opportunities to combat pain points, but without good practices, they are not a full solution. So that leads to the question of, we need to combine best practices to empower the existing tools, making sure that we are using best practices for FAIRR at every level. This will allow you to maximize your analysis and make it fair and long-lasting. Your code will be optimal now, and it will also be equally optimal 5, 10, 15 years from now with the FAIRR tech stack. So now we can actually get into building your tech stack, or more importantly, the practices that can be applied at every layer with the existing tools we have. So let's begin with environment. Environment, in this case, we are going to employ the tool of Docker. We want to ensure that all the packages we are using are operation system agnostic. This can be checked in package managers like Conda, ensuring that the package can be accommodated for all architectures available. More importantly, as I highlighted previously, for all the versions of the package, for all the packages that you install in your Docker container, it would be beneficial to ensure that the versions are pinned. specifically, once again, with variable packages like TensorFlow and PyTorch, where the versions are subject to change, and that can lead to function changes over time. And lastly, it is important with an ongoing Docker container to ensure that any out-of-date packages or images that have been deprecated that you are using in your container are replaced. This can lead to polling issues if end users try to replicate your original environment. If a package or an image is no longer available, that can cause some issues. So establishing the best practice we have for environment, how can we add on the code? Code, as we established, we are going to store in GitHub or a Git provider. So the environment or the Docker file that we are using to build our custom environment and our code can be both present on GitHub. Docker files or the file necessary to build a Docker container is relatively lightweight. It's a text file, so it can easily be stored on GitHub along with the entire code base. When it comes to providing your code base on a platform like GitHub, your code should be updated to maximize reproducibility and execution. This might include making your code compact, or making your code explainable, or maybe setting a designated run file to ensure your code can be run end-to-end with a single command. And lastly, to ensure users can run it regardless of their programming knowledge, it's always important to include comprehensive documentation within your code base on GitHub. This might include very typically like a README file or some other wiki files within GitHub to provide comprehensive instructions on what the code does, how to run the code, and how to alter the code for reuse potentially. So now we have the best practices for environment and code. We can add in data. We now have environment code on GitHub, both following best practices. When it comes to data, this would vary depending on the source. If it's from a public repository, a potential practice you can employ is storing the data as a backup on a long-lasting source site for which you have ownership. If it was originally present on Geo, SRA, Zenodo, where you do not have ownership, it could be deleted without your knowledge. And so it's really important, or it could be a good practice, to not rely on an external repository. Maybe store a version of the data on S3 or through another direct download method. The link for accessing the data could be put in the GitHub that houses the code and environment, and any relevant metadata, including organism, data type, data file type, method of extraction, should be included as well in the documentation that was made for the code. This might include a README file or another text file, some descriptive information about the data, so that reuse with new data files is also seamless when it comes to the end user. So now that we've kind of gone over the best practices in the FAIRR tech stack, I would like to present you a case study, specifically centering around FAIRR and computational tools. The example I would like to give is in reproducing an original study. Let's say you are beginning with the publication on the left, which is Scampi of large-scale single-cell gene expression data analysis. It comes from the Phase Lab, and it's a very popular toolkit to use for functionality similar to Seurat and DESeq2 in Python. In the middle, you would have all the necessary components that you were able to find, including code, data, and environment. And on the right, you have a UMAP plot by Leiden, by the Leiden algorithm, to show differentiations in cell expression. So you have this typical process that you would see when it comes to reproducing an original study. And the original author has set it up to get you this plot that you see on the right. So what would happen if we tried to reproduce it? And more importantly, what would the result be without the tech stack? Or what would the result be with fractions of the tech stack present from the original author? And we can look at this component by component. So let's say when you were beginning the reproducing, the code and the data were following best practices. You were able to access the entire code base incredibly easily. You were able to access the data incredibly easily. And the issues came at the environment. We can see different patterns. If the tech stack was used, the code would be present, the data would be present, and the environment would build smoothly. What would that result in? It would result in the exact same UMAP plot we previously saw in the original publication. What if they were using the tools but not the tech stack practices, specifically in the environment? Maybe the original author used Docker, but the version of a package was not pinned. So when the Docker image was pulled 5, 10, 15 years from the original publication, the version had been updated and the function had slightly been changed to make the UMAP plot. You may see a similar UMAP plot, but you will see different contents that will not validate the original plot of the study. And lastly, if no tools or tech stack practices were used at all, the environment would not build. And so what would it result in? It would likely result in an empty plot because you were not able to run the code and the data, even though they were present in their best practices. We can also see a similar pattern with the code. Let's say environment was built correctly using best practices, no issues there, and you were able to find the source data easily linked. When it comes to code, if they were using the tech stack, all the code base was available on GitHub. It was designed to maximize reproducibility, and it worked as expected, which results in the exact same reproduced UMAP plot as in the original study. If tools from the tech stack were used but not the practices, all the code base may be available on GitHub, but the running may be cryptic. So in a Scampi example, maybe you followed the use of the wrong data set, or you followed the wrong pre-processing options. Once again, that might result in a slightly different UMAP plot that does not validate the original findings. And lastly, if no tools from the tech stack nor practices were used, the code would not be available, or the code would not be runnable, which results in an empty UMAP plot. And lastly, we can see this exact same path with the data. If the environment is built with best practices and it builds correctly and the entire code base is available for runnability, we can see some variations in the data. And this is something that I've personally experienced in my graduate work and also some other work I've done in research. When it comes to using tools and practices from the tech stack, data will be processed and sourced easily with the identical code and environment that we previously had, and we can result in the same UMAP plot. If tools but not tech stack practices were used, the data may be linked in the GitHub from a geo or Zenodo link, but there may be identical names and versions of that data. So you won't be able to determine what is the correct choice. Maybe you select a data name or data file that has the same name, but is actually a subsetted version of the original source data used in the original study. That will result in a different UMAP plot. And maybe without using tools or tech stack practices, data can't be found at all. It's only available upon request, which isn't answered, or it's only available behind a paywall. And that unfortunately results in an empty UMAP plot. It's important to highlight that data, sometimes there are certain regulations that are put into place, which is definitely understandable. But in this case, we're thinking about publicly available data that was not linked. And so now I would like to do the user poll after going through that case study of which of the following do you think is most important for authors in this specific context to provide for their code to be reproducible and reusable. We can see what everyone thinks about the poll. Okay, perfect. We've just ended the poll and it seems like a lot of the audience has voted either between having a pre-configured environment and having all the components of the necessary analysis present, which is definitely components that are difficult to replicate if you do not have prior knowledge. I see some other audience members have also voted for providing a path to the correct original data, and I definitely sympathize that all components are necessary. And it's important to note that practices that could result in disadvantages or disadvantages with using specific tools in the tech stack, they don't always exist in harmony. Maybe only the environment has a pain point, or only the code, or only the data. It doesn't necessarily mean all of them will have pain points in their own right. If we could go ahead and end the poll, I can move forward. Perfect. I actually wanted to give a personal example at the end of this case study. As part of my graduate work, I wrote a reusability report in Nature Machine Intelligence on this very topic. The original study I was doing it on was from the IDGR lab in Nature Cancer, and it was evaluating a fine-tuned model to predict drug response in cancer patient samples trained on large cell line data sets like GDSC, GCSI, and CTRP. And it was training to determine whether drug response with respect to continuous IC50 values could be predicted in patient-derived xenografts. And so I did some evaluation of the reproducibility and reusability which I felt was really important to highlight the different paths we can see when it comes to variability of environment, code, and data performance, because I definitely experienced variability in all those components, which kind of led to my appreciation when it comes to a full end-to-end workflow abiding by FAIRR and open science. And so now that we kind of talked about the tech stack and the benefits and the best practices that have to be employed at every level, I wanted to quickly go over how CodeOcean employs the tech stack, because you might be thinking in the audience, well, how can I actually start using this tech stack in my own work, even though I have existing projects? And I wanted to highlight how CodeOcean was able to do it. So in CodeOcean, all of your code is accompanied in a compute capsule via built-in tracking with Git from start to finish. And this is available via our timeline. You can easily track your code start to finish with a built-in Git UI. In CodeOcean, our sharing system for collaboration prevents merge conflicts and prevents synchronous user work on the same project, which allows you to avoid any changes that might result in merge conflicts, and also allows you to track changes unique to the user. Also within CodeOcean, to optimize the execution of your code, we have runnability options available by different user type. We have both code and non-code running options, depending on your collaborators. When it comes to storing and accessing your data in CodeOcean, there is data persistence. When data is imported from different sources, it has to be explicitly removed by the original author twice in order to actually be deleted from your library. In CodeOcean for data, we have certain capabilities available for metadata configuration, which allows you to automatically, when creating a data asset, provide detailed information about the data source. Once again, this might include organism, data type, things like that. And also when it comes to data, we have provenance capabilities available. When generating a result from a given analysis workflow, there are very specific provenance capabilities that are available, including the code that was used to run it, the commit version that was used to run it, the run file, maybe some parameters that were used, things like that. And lastly, when it comes to environment, in CodeOcean, we have an automated non-coding UI for seamless configuration of Docker images to use as your cloud computing environment. We have preservation of the original Docker environment layers via automatic package versioning from computation to computation. And reproducibility is preserved with sharing between collaborators, as collaborators will work on the saved build states that were previously used in the last computing session. So essentially, the pain point that we previously experienced from the audience of it works in your machine but not mine is removed, as users will be working on the same operating system, the same machine, with the exact same environment, package specific. And so now we can do a summary of what we discussed today in this webinar. We established there are known pain points in life science computational workflows that impact the path from data to discovery. To combat this, there are several available tools to actually empower bioinformaticians or computational biologists. Platforms like GitHub and Docker allow scientists to make their analyses fair, reproducible, but unfortunately there are certain habits with these tools that aren't usually highlighted but can actually sometimes cause more harm than good, as we previously highlighted with maybe packages going out of date or code bases in GitHub not being set up to fully maximize reproducibility. And so the question we began to ask in this webinar was, how can we use these tools to their best ability? More importantly, how can we use them to build a fair tech stack? A tech stack we defined as using multiple tools at once in a layering format for their full capabilities. You saw when I was building the tech stack, as I added a layer, I was incorporating elements of the previous layer to ensure that they were all connected. You want to ensure you're building best practices at every level and ensuring your code will work now at an optimal level and continue to stand the test of time, which could lead to a very reproducible and reusable analysis, which is ultimately very beneficial when it comes to computational biology.